Okay, everyone, we are live. Welcome back. It is another Thursday afternoon biotechnology live stream. We are happy to be here with you today. It's a lovely afternoon in Maryland. My name is Danielle Snowflack, and I'm the Senior Director of Education at Edvotek. We're happy to bring this biotechnology training workshop to you in the comfort of your own home. One question I have for you all who are attending is if you let us know where you are located. We're starting to think about what time to schedule these workshops during the school year, and we want to know what time will work best for you. So in the comments, please, in the live chat, please let us know, or in the comments that come along with the slide request form. So, we want to make sure that the timing works best for you when you are in the classroom. We're going to talk about another biotechnology concept today. We're going to be talking about chromatography. And I know a lot of people think of Edvotek as the PCR and electrophoresis company, and they're very popular experiments, and they're robust and they're affordable, and students really love them. But we have a lot of other biotechnology experiments that we have to offer, and I'm going to run through one of them with you today. So for those of you who may be new, let me advance the slide. Go back. For those of you who may be new, we are Edvotek. We are the biotechnology education company. Hopefully you know a bit about Edvotek by now if you've been on some of these live streams with us. But we were founded as the first company to bring biotechnology education to high schools. We were founded over 30 years ago by Dr. Jack Churchian, a professor of biochemistry at Georgetown University. And I always like to just mention how exciting, the world of biotechnology was about 30 years ago. We were making huge advances with the Human Genome Project, in learning about DNA sequencing. We were starting to use the polymerase chain reaction, or PCR, in the laboratory. Genetic engineering became popular, and that allowed us to do experiments like producing insulin in bacteria, which is a medication that's critically important. But little of that was being translated into the classroom at the time. And so with that, Edvotek was born. Our focus is solely on bringing these experiments to the education sphere. We work with educators all over the country and all over the world to help them demystify science and to foster the next generation of scientists through hands-on active learning activities. And so today's activity is a STEM activity. And STEM is one of these, and this experiment is a chromatography experiment that we will be doing to separate different dye molecules by their charge. Chromatography in general is the separation of molecules based on their physical properties. And this is a really important process for many types of industrial research and development, including in the biopharmaceutical industry, which is what we're going to discuss in this workshop. The experiment we're running today is Edvotek Kit 243, which is our ion exchange chromatography. In choosing this experiment, this chromatography experiment, we are going to dive into a lot of STEM concepts. This is a really natural experiment for STEM. We are going to separate these brightly colored dyes by their charge. We're going to talk about engineering of these columns. We're going to talk about technology for protein analysis, just a lot of different aspects of the process that one would go through in doing a chromatography experiment. And then we're actually going to do a mathematics extension activity, which involves measuring the absorbance of dyes to calculate the concentration of our products in each fraction. And then using this information, we can figure out how efficient our separation was. So we are recording this demonstration, and the slides will be available on our website. If you'd like to be notified when they're posted, please fill in the form. The link has been posted in the chat box, so you can just click on that form and let us know your name, your email address, and we'll send it to you. In addition, we are also offering professional development certificates live to those people who are watching. If you want us to send you a certificate, please be sure to check the box to let us know to send it to you. We don't want to send you, materials you don't need. This link will only be live for 1 hour after the presentation, so be sure to complete it before 3 p.m. Eastern Time. So let's start talking about STEM. What is STEM anyway? And again, this is one of these buzzwords in science education that's been around for a while. STEM lessons are supposed to be comprehensive lessons that integrate science, technology, engineering, and mathematics into an interdisciplinary unit. STEM breaks down complex questions, challenges, and problems, or global issues into parts to discover the nature and relationships between these problems. This involves reading and researching not just the hard scientific literature, not just biology, chemistry, physics, but also sociology and psychology and the human implications behind a given topic. Using this, we then identify and understand the content knowledge from STEM and other disciplines and use a line of reasoning to generate necessary solutions to real-world problems and other complex questions. But in my mind, I call this good science. So STEM is a buzzword for doing what we should be doing in the lab anyway. So biology itself does not exist in a vacuum. For example, my training is in molecular biology, and I worked in a fruit fly development lab. One thing we did was to look at fluorescent tag proteins in fly eggs, which help us learn about gene regulation. So the way we go from the sequences in our DNA to the proteins that are doing the work in our bodies. And so we had to use a lot of different concepts. We used genetic engineering to make the recombinant DNA molecules that were then injected into the fly. Once the DNA is spliced into the fly genome, we would use mathematics to calculate the probability that our protein of interest was in the different eggs. So if a parent fly has one copy of the gene, we have a 50% chance that it will be in their offspring based on meiosis in the eggs and in the sperm. The results from these, and then to look at these fluorescent proteins, we need a very sensitive microscope to actually be able to look at these microscopic, tiny fluorescent particles in an egg. So it's an amazing piece of technology that lets us look, at minute amounts of protein in a cell. So the results of these experiments helped me to understand how genes are regulated and expressed from DNA to RNA to protein in cells. And that's important for human health and human biology and health and development. Misregulation of gene expression causes diseases like cancer or osteoporosis, and there's a lot of different diseases that can be linked to protein misregulation of gene expression. And so this is an experiment of a good experimentation. So we're using science, technology, engineering, and mathematics. They all depend on one another, and they all build into a comprehensive experiment. It's just a matter of making sure that we can identify and discuss these different topics. So biotech is actually great to teach STEM because these experiments really integrate science, technology, engineering, and math to solve real-world problems. The important part is when talking about these interdisciplinary experiments is that the students do learn to make connections between the STEM concepts in the classroom and its practice in the real world. So today we are talking about chromatography, which is a very important technique. Just a little bit of history. The word chromatography is from the Greek words chroma and graphene, which roughly translates to color writing. And so something really interesting that I learned in doing the research for this workshop is that the botanist Mikhail Tzvet, and I'm sorry, I'm probably saying that wrong, who was responsible for the first real chromatography experiment, named the technique after himself. So his last name, the Tzvet, T-S-V-E-T, basically translates to color in Russian. And so by using the Greek chroma, which also means color, he may have been naming the technique after himself. So the precursors of today's chromatography can actually be traced back to the 19th century, where researchers wanted to analyze the pigments in plant leaves. In these experiments, scientists crushed up leaves, mixed those pigments with solvents, and spotted them onto filter paper. As the pigments traveled through the paper, they separated into distinct pools, creating bands with different colors. And so here is actually an experiment. In this picture, you can see this is a modern paper chromatography experiment where we're separating different colored dyes. It's a simulation of analyzing the ink found on a ransom note in a crime scene. But basically, we spot the different colors onto the paper, and we use the flow of a solvent through the paper to separate the different molecules into distinct fractions. but they were just kind of spotting them in the middle of a piece of paper like a dot and just letting them spread outwards instead of spreading upwards in an orderly process. So in the very early 20th century, Svet applied the principles of paper separation of the plant pigments to column chromatography. I couldn't find a picture of that. This is a picture of chromatography in the 1940s. You can see these are gigantic columns, larger than the researcher himself. But for these initial chromatography experiments, calcium carbonate was packed into a column and used to separate the pigments instead of separating them on paper. Furthermore, Svet realized that the solvent used for chromatography was important. So certain molecules would separate better when separated using polar solvents versus non-polar, and vice versa. And this is all dependent on the nature of the molecules themselves. So in the mid-19th century, researchers continued to expand on their knowledge of chromatography techniques to include many different forms of chromatographic separation. So we're doing ion exchange chromatography today. That's going to separate molecules based on charge. What we see in the picture here is an experiment that we offer, which is size exclusion chromatography, which you also may think of as gel filtration or molecular sieve chromatography. And so this is going to separate molecules based on their apparent size. We have We have affinity chromatography, which uses protein binding interactions to separate molecules. So we have a binding of an antibody to an antigen. They fit together and they stick together until we elute them using the appropriate chemicals. And then we also have something called thin layer chromatography, which instead of packing the matrix into a column, we actually spread it in a thin layer on a sheet of plastic or glass to do the separation. Furthermore, we can talk about engineering when it comes to technology that we use to do chromatography in the lab today. So what we're looking at here is a fast performance liquid chromatography machine, or FPLC. Engineers developed this machine to perform chromatography under pressure. This allows a researcher to control the speed at which the liquid is flowing through the tube instead of relying just on gravity. We're going to rely on gravity today, though, because this is a $20,000 piece of equipment easily. And, you know, that's for the entry level. And so gravity is free and based on our Earth. So where does HIC fall into those four? So there's a couple different things I can think of as HIC. Can you just spell out the acronym for me, and then I'll fit it in. It could also be I just picked a couple examples, and it could be that I didn't necessarily put it into this particular breakdown. You know, there are more chromatography methods that aren't listed. Oh, hydrophobic interaction chromatography. Okay, so that's going to be a little bit different, but it's going to be similar to ion exchange, where we are using the physical properties of the interaction between the matrix and the protein. So for hydrophobic interactions, we're going to play on hydrophobic versus hydrophilic. So you would imagine that anything that is hydrophilic will be able to flow through the column very quickly. Hydrophobic will bind to the resin, and then we have to elute it in the proper solvent. So that's a great question. I put these four in the slide. Again, these are also going to be a little bit easier to do in the high school classroom. Hydrophobic interaction chromatography might need to be done with harsher chemicals that can be done in the average high school classroom. So great question. Thank you for asking. So let's talk about what we need to perform chromatography experiments. I am going to now switch to my camera view so that we can see what's going on all together. So what you're looking at here is the column. This is my basement setup. So if you were in the lab, you doing a gravity chromatography experiment, the best thing to do would be to use a ring stand. But this is a simple and easy fix. I'm just using a clear plastic cup with the side cut out and a hole poked into the top. So what we're looking at here already is the chromatography column. This column is a tube with a frit and an elution spout fitted at the bottom. The frit is a membrane or a porous disc that supports and retains the matrix in the column, but allows water and dissolved solutes to pass through. We're going to pack this column with our ion exchange resin. I'm actually going to pack it live for you. So you can see here is our resin. It looks cloudy now, but when we pack it into the column, it will settle into kind of a white layer in the column. You can see I bumped my camera from where it was set, but that still looks good. So this resin is an ion exchange resin, which consists of microscopic beads, and these are going to be charged. So in our case, we're using CM cellulose, and this cellulose has a negative charge. The matrix is going to capture molecules that pass through the matrix. It's going to be able to interact with positively charged molecules. We're going to be using buffers. So I have a big jar of buffer here. This is going to be my low salt buffer, and I also have a high salt buffer. And we're going to be using those to do our elution. I have a dye mixture. In this case, we are using a mixture of 2 different dyes. And you can see that this is kind of a sludgy brown color. And just by eye, we can't tell if this was one molecule that was brown or a mixture of 2, 3, 4, however many different dyes that could come together to make this color. It's pretty dark. You can imagine more than 2 dyes that were put into this tube to make that color. And so using the physical difference of these dyes, in this case, the charge, because it's ion exchange chromatography, we are going to use these properties to separate them. So we're going to transfer our samples and buffer. So I have a collection of transfer pipettes. These are plastic transfer pipettes and an adjustable volume micropipette. So the plastic transfer pipettes, are simple and easy to use in the context of the basement. We can use either plastic transfer pipettes or the micropipette, which is a precision tool that is used to make measurements of very small samples. I'm actually going to use both today. I'm going to use the transfer pipettes to measure the larger quantities of buffer, but I'm going to use my adjustable volume pipette to make sure to add a precise amount of my dye sample. And then we're going to elute the samples into some microcentrifuge tubes. These are essentially mini test tubes that we're going to use to collect the fractions as they come off the bottom. All right, so here is where the experiment is going to happen. I am going to start trying to pack this column. This can take a little while sometimes, so I'm hoping that it goes quickly. If not, I always have a backup, so I have a backup column prepared. But basically, we are going to fill this column with our resin. We're going to allow the extra buffer to drip off, and we are going to, so we're going to basically have all of our solid matrix in our column. Now, there are a bunch of other steps involved. We would need to rehydrate the matrix. It goes through several steps of washing, and if we did that, we would not have the time. But if you want to see that, we do have a video on our YouTube channel that will go into the details of packing the column, preparing the resin, and any of that information that you might need. So what you can see is that we have this cloudy mixture going into the tube. And what you'll see is that the buffer is flowing out the bottom, but the cloudy resin is going to stay inside the tube. While this is packing, I'm going to talk a little bit more about chromatography. We want to make sure to keep an eye on this column so that it doesn't dry out, because that would be a problem. So I'm going to be going back and forth between the column and these slides. So chromatography techniques separate molecules based on their physical properties. This is ion exchange. It's going to do positive or negative charge. In the case of hydrophobic interaction chromatography, which we asked about before, it's going to be whether the molecules are hydrophobic or hydrophilic. Here we have the column that we're packing with the matrix. The matrix is known as our stationary phase. The packed matrix is called the bed, and the volume that it occupies is called the bed volume or the column volume. It's very important not to let the bed run dry, otherwise cracks and fissures are going to develop and the matrix might have to be removed or repacked. So you can imagine if we have a bunch of channels in the resin, our molecules could get around these channels instead of having to go through our beads and our resin. All right, I'm going to add a little more just to keep things going. So the matrix is used to physically separate the molecules into different pools based on their physical properties. The elution phase is our mobile phase, the mobile phase of chromatography, and that's what's flowing in through the matrix and out of the column. The column with the matrix and the applied sample is developed essentially by the elution buffer. This means that the molecules in the sample are carried by the flow of the buffer into the matrix where they are gradually separated. The separated zones of molecules then flow out of the column where they are collected for analysis. So let's look at an elution profile and define some more key terms. So when we're looking at this picture, the peaks are going to represent the partially or completely separated zones of molecules that are eluted in the column. So each of those numbered peaks, 1, 2, 3, 4, 5, is going to represent a separate elution, a separate molecule, or a mixture of molecules that happen to flow out of the column at the same time. The total volume of buffer between the beads is called the void volume. And this is going to be representative of whatever flows through the column without interacting with the beads. So in ion exchange chromatography, any molecules in the column load that do not electrostatically stick to the resin will elute in the void volume, since the charge is our major parameter for separation. By contrast, molecules that stick to the beads will be fractionated based on their charge. And so then we also have the bed volume. So the bed volume is going to be the volume of the beads plus the void volume. So that's basically the summation of everything that is in the column, that is going to be in the column. And this is going to help us make calculations for how much buffer to add when we're doing chromatographic separation. So for me right now, I'm kind of winging it. I'm just kind of adding it and I'm eyeballing to see if we have the right amount of molecule of resin in this column. If you were actually in the lab, you would want to more carefully calculate it and add the correct amount, simply so that you get, so that when you do your calculations for how much buffer to add, how much sample you're going to get, you have more precise numbers. All right, so our column is pretty well packed. What I'm going to do now is I'm going to remove this extra buffer from the top. I'm going to try and remove most of it without disturbing that top layer of our resin. I'm going to leave a little extra because it's going to continue dripping for now. So here we have our column. So what we want to do, first step would be to label, after we pack the column, we are going to, now let me do this. So now our column is pretty much empty. The buffer's pretty much run out of the column. The only buffer we have left in here is going to be our bed volume. So the volume of the void volume, the volume of liquid that is in the bed. And then we're going to be, I pre-labeled them, but you're going to want to, at this point, you're going to want to label your sample collection tubes. And so to be able to hold the tubes while we're doing the experiment, I actually have a little bit of this dough that children play with, just for copyright infringement reasons. I won't use the name, but if you've got kids around, you probably have some of this in-house. And I'm actually going to use this to be able to hold my samples under the column as they elute. So this is our little setup. It's not as fancy as you all probably would have in your labs, but this is basement biochemistry. So, we make do with what we have. And again, this is just a plastic cup with a side cut out of it. So I've removed the excess buffer. I'm going to take my micropipette and I am going to very carefully load my sample onto the top of this buffer. All right. So we want to do this very slowly and gently. I'm going to put the pipette on the side and just gently drip this on. And you can see it's going to form a nice layer on the top of the column. All right, eject the tip. Now it's loaded. So next I loaded the column. I'm going to remove the cap. Oh, actually, I want to go back to my beaker here. We're going to let the sample enter. And then we are going to add some of this low salt buffer to the column. And I will further explain, once this is loaded a little more. So we're just going to let that load. I'm going to leave this pipette here for now. Hopefully it doesn't fall all the way in while that's loading. I'm going to let this load for a couple seconds. I'm going to move to the next slide, but we will come back to this because I want to talk a little bit about what is going on with the molecules. So the chemical principles of the chemical principles behind this chemistry. Ion exchange is going to be based on the charge. Does that look like it's loaded? That looks like it's loaded. So let me add some of this. This is our buffer. Oh, thank you. I'm glad you like the idea of the cups and the clay. It's nice to have the best of everything, but in the expensive ring stands, but you don't need fancy stuff to do biochemistry in your classroom. Talking about the compounds, most biological compounds are positively or negatively charged in solution, and this is dependent on the chemistry of the molecule. So if we're thinking about a protein, amino acids have side groups like carboxyl groups and amine groups that can gain and lose proteins depending on the pH of the solution. This affects the overall charge of the protein. So just by changing the pH of a solution, the net charge on a molecule can change from a neutral to a net positive or negative charge. The isoelectric point of a protein is the point where a molecule has no charge in solution. So the pH equals the pi. So knowing the charge of a protein, let me get my sample collection tube under here. We're almost, if you can see, we are almost at the point where the red dye is about to exit the column. So I am going to put my R1 fraction under here. Let me get that lined up. So we have R1 and we are starting to collect the fraction, which is exciting. I also have little lines on each of these tubes, which is where the 1 mil mark is, just so that I know when to change the samples. And you can see the red dye is flowing off the column pretty quickly. So knowing the charge of a protein is important in ion exchange chromatography because the technique is based on electrostatic interactions. So basically, the fact that opposite charges attract like a magnet. So for example, today we're using the cation exchange resin, CM cellulose. This negatively charged resin will capture positively charged molecules. So now if we had a protein with a pI of 7 in a high pH solution, our protein would likely have a negative net charge and not stick to the matrix. So we would want to have a low pH solution under 7 so that our protein has a net positive charge and would be effectively purified using the ion exchange chromatography. So we can easily calculate the pI of a protein by looking at each of the amino acids. So these calculations can be done by hand, but luckily there are bioinformatics programs that take care of that for us today. So I'm going to just walk through a program called Uniprot, which is from the online bioinformatics resource portal, which is freely available online. And so I'm going to run you through the process of calculating the pI. Actually, first I'm going to change out this tube. You can see I'm missing a couple drops. In an ideal world, we'd have one person doing the talking while another person is doing the experimenting, but we're improvising here in my basement lab. We can easily calculate the pI of a protein looking at each of the amino acids. And so again, this can be done by hand, but we have programs that are going to take care of this for us. So I'm going to run you through the process of calculating a pI. Is that dripping in there? Yes. Okay, so first you would go to the website, which is xposi.org, and you would, in the top drop-down menu, select Uniprot KB. You're going to insert your gene of interest into the search engine. So in my case, I'm looking for the fly protein glorand, which I worked on in grad school. Hit search and you are ready to go. So we're going to get a list of hits, and in this case, it's the first one. And we're going to click on the entry to bring up a wealth of information about the gene and the protein. So next slide. Come on. All right, here we go. So here we are looking at the entry. So if you're a biochemist, I highly recommend spending some time with this site and the information. However, we're going to just move right down to sequence. In the sidebar, you'll see another drop-down menu. If you select program and hit go, the software will use the protein sequence to calculate a lot of information about the protein. And what you'll see, let me add a little bit more buffer to the top here. Or actually, let me cap this for a second while I finish this next little part of the lecture. And then we'll go to eluting the next color. Okay, so as I said, we have a wealth of information about the protein. The program examines the amino acid sequence, amino acid by amino acid, to calculate the molecular weight of the protein, the percent of each amino acid present, and importantly, the PI of the protein. So in this case, Glorant has a PI of 5.71, meaning that at the pH of 5.71, this protein would be neutral. It would have no charge. So as a complementary activity in your classroom, you can have your students identify a protein of interest. Either you can assign a few or they can choose one, and they can calculate the PI of the protein using the software. You can use that as part of a lab report, or you can use that as part of a lab report, or you can have the students present it in class. And so these are real tools that are used by researchers in the lab every day. So the protein number, I'm looking at the picture. So what I can see is that, we have accession numbers. So the accession number is largely a reference number. So it might be reference to a database where the sequence is held. Some of them will link to the protein sequence. Some of them will link to the DNA sequence. It's going to be a little bit different for each one. But what you can see is that we have that identification number. We have the identification number, the AT number. My guess would be that that is related to its DNA sequence, where the Q9VGH5, I believe that is the accession number for the protein or the protein structure. What is the number for the specific protein that we're looking at in the chromatography experiment. So we're actually using dyes here. And I don't have the exact charge of these dyes in front of me, but I can look that up and put it in the notes. All right, so performing ion exchange chromatography. So we have eluted all of our red, and now I'm going to close this up because we are going to go to elute the blue. So this first buffer was our low salt buffer. So that is going to wash away anything that's on the column that doesn't have a strong affinity. Now, by using the high salt buffer, we are going to wash away anything that has a strong affinity for this resin. And so, again, I'm going to be getting a little bit ahead of what we are doing versus what we're talking about, but the next little discussion will talk about the differences between the high salt and the low salt buffer. So let me add this to the top. And we want to do it very gently so that we're not disturbing our matrix, our bed. I have the column right here. I have the column capped, but we're filling it up. I'm going to put my sample B1 in and we'll go from there. So what type of biomolecules can be purified with ion exchange chromatography? I mean, we could do DNA, RNA, no problem, different metabolites. Those would all be able to be separated using ion exchange chromatography. And not just ion exchange, I mean, many different chromatographic separations can be used, can be applied to biomolecules. So, and then also non-biological molecules. So, chemical compounds are separated. These are dyes, they're organic molecules, and they're easily separated in this way. While these are eluting, I'm going to keep talking. So ion exchange chromatography works because we're taking advantage of the fact that opposite charges attract. So basically, the ion exchange matrix is going to have either a positive, negative, or positive charge. Again, today we're using the cation exchange resin CM cellulose. This means that the resin is negatively charged and it will capture positively charged molecules. We loaded the sample onto the top of the resin and we let it enter. Then we flow the buffer through the column to elute the different dyes. Any negatively charged molecules will elute first, oftentimes in the void volume. That's what we just saw. This is because they don't stick to the charged matrix. Negative molecules are going to repel each other, and neutral molecules are not going to stick to the matrix. So they're going to elute in the early fractions. All right. All right, let me Move this around so that we get our next fraction going. Again, this is much easier if you have someone to hold with you and work together, but that's what we're doing in the lab. Your students will be working together to elute these molecules. We talked about negative and neutral molecules. Now, positively charged molecules are going to stick to the CM cellulose resin. To elute these molecules, we change from a low salt buffer that we use to load everything to a high salt buffer. So we can elute proteins either by changing the pH or by changing the salt. The salt is going to change the way that the positively charged molecules can interact with the resin, with the negatively charged resin. Molecules with weak positive charges move out of the column more quickly than those with high positive charges. What is that? I need tube 3. We are getting pretty full. There's tube 3. So molecules with weak positive charges will move out of the bottom of the column more quickly than those with high positive charges. Basically, the more charge you have, the stickier a molecule is, and the harder we need to work to get it to stop binding to the matrix. We can collect the eluate at the bottom of the column in separate pools that we call fractions, and each of these fractions is going to have a different concentration of our molecules present. All right, let me change this next fraction. What you can see is that the samples are getting to be much darker in blue color. And we can see that the resin itself is starting to get less blue as the blue dye moves through the column. I hope you can see those differences. All right, so let's take a look at what our elution profile might look like in an ion exchange experiment. So this is a step elution, meaning that we change the ionic strength based on the salt concentration in regular intervals. And so that's what we can see on this graph as the red line. So CV means column volume. Again, that's the volume here we have of the matrix plus the buffer in the column. So if we look at the blue line, that's our elution profile. And you can see that there are several distinct peaks where molecules pass out of the column. In the early fractions, we're going to have negatively charged and neutral molecules passing out of the column. They pass through quickly because they don't interact with the resin. So once we've washed away the molecules that don't interact with the matrix, we change the buffer to elute the bound molecules. Let me add a little more buffer because we're starting to get low. And so again, you can have one student collecting the fractions, one student can be adding the buffer, one can be collecting the fractions. So it's a good activity to do in groups. I'm going to get the B4 tube out. And we are going to start collecting the next fraction. Next time I'll cut a slightly bigger hole in the cup to make it a little easier to maneuver my hands in there. Okay, so once we've washed away the molecules that don't interact, we change the buffer to elute the bound molecules. This is done by either changing the pH of the solution or by increasing the amount of salt. The pH is going to affect the charge of the protein, as we discussed before, by changing the charges of those side chain molecules. If we change the concentration of salt, we're actually introducing competition for binding to the column. By increasing the concentration of salt, the positively charged salt molecules, so in this case we're using potassium acetate, so the potassium molecules, are going to bind to the negatively charged resin and displace the positively charged dye molecules. The separation of compounds is based on an equilibrium of molecules bound to the exchanger versus the elution buffer. So the equilibrium can be shifted gradually by changing the ionic strength, which we're using here, or the pH of the elution buffer. All right, let's get this next fraction. I'm going to take this one out and start doing this next fraction. All right. In this experiment, by adding a lot of salt, we are weakening the electrostatic forces between the molecules in the matrix, which is going to elute the molecules from the exchanger. So basically, the charged salt molecules kick off the dye molecules. Molecules that have a weak positive charge are more affected by the changes in the buffer, and they will emerge first, followed by those that have a strong positive charge that bind more tightly to the resin. Because we can control the concentration of salt, we can control the way these dyes elute. In this example, if we're looking at this graph again, we can see that our target molecule elutes at a higher concentration of salt than the unwanted material. And this is going to allow us to separate molecules with small differences in net charge. Can you still see that? Let me adjust that a little bit. This little flag should be facing forward so that I can get that column better aligned. Okay, so industrial applications of chromatography. So when do we use this? So in the pharmaceutical industry, many drugs are produced in living cells in a process called fermentation. For over 6,000 years, fermentation has been used for food preservation. So if you're thinking about food products that are fermented, this is a natural process from microbes. In the 1950s, researchers learned that fermentation is actually the process of microorganisms breaking down the chemical bonds from sugars and starch molecules into different products like lactic acid or ethanol, acetic acid, which are going to be, and breaking these bonds actually produces energy for the bacteria. Now, the byproducts of this process, especially when they're in our food products, are going to produce delicious foods that we love. So you could, foods like kimchi, yogurt, sauerkraut, wine, beer, all of these are produced by fermentation. But current technologies have expanded the utility of fermentation. So we can actually now exploit the process of fermentation to manufacture products as diverse as biofuels, biopharmaceuticals, and fine chemicals. The process of growing and purifying industrial products in cells is known as bioprocessing. Upstream bioprocessing uses genetic engineering and recombinant DNA to produce large amounts of important proteins, essentially converting cells into living factories. Now, once fermentation is complete, the desired molecules must be harvested and purified from the culture. This practice is known, so basically this whole process is known as bioprocessing. Once the fermentation is completed, the desired molecules must be harvested and purified from culture. This is actually known as downstream bioprocessing. And the product, which, if we're talking about something like insulin, which is a medication, is going to be purified from the cells using centrifugation, filtration, and chromatography. When purified, the product can be utilized for commercial or industrial purposes. All right, so that was our last fraction. So let me cap this column. This actually went more quickly than I expected, which is great because in some of my trials it took a little bit longer. We're doing real science here, and sometimes it's not exactly, it doesn't exactly agree with what we intend. But I have a couple more slides, and then we're going to talk about the results. So when we purify these different proteins from these crude extracts of broken cells, we're going to centrifuge it, we're going to filter it, we're going to purify with chromatography, and then we can use this product for commercial or industrial purposes. So insulin, which is a hormone used to control diabetes, was actually the first medication for human use that was produced by genetic engineering and purified in this way. And today, biologic medications like antibiotics, interferon, and blood clotting factors have helped to save millions of lives and improve the quality of life for millions more. So, since chromatography is an essential step in the production and purification of pharmaceuticals, there are many positions open for scientists with experience and knowledge of these techniques. For example, in the laboratory, we have downstream bioprocessing scientists who are responsible for the purification and characterization of manufactured chemicals and biologics. They are going to design and execute separation techniques that are used in the laboratory. Chromatography specialists are experts in separation processes. They will consult on the design of separation experiments, and they may also work as field technicians, traveling from site to site, repairing and maintaining the equipment necessary to perform chromatography. The quality control manager is an essential part of the manufacturing of drugs, reagents, and other chemical products. They review data from chromatography experiments to make sure that they meet established quality standards, and this is going to mean that there are no errors or misinformation. This ensures that experiments are done to standard, resulting in a safe end product. So if we are creating biological medications, we want to make sure that the medication is going to be safe, not only in testing it to make sure that it works for the intent of the medicine. So if we're talking about insulin, if it helps us control diabetes, we also want to make sure that the process itself doesn't leave any contaminants behind that are going to hurt people. All right, so let's talk about our results. Now we're caught up. So What you can see is in that picture, you can see some characteristic results. I am going to adjust my setup so that you can see the fractions that we collected from this experiment. My table is like a light tan color, so there is going to be a little bit, the color is going to look a little different than it should, but it's going to be a little bit different than you would see, at home if you were doing it on your, on your lab bench, just because the colors are not reproducing perfectly. Let me angle this arm up a little bit. So we did get a question, where can you get these experiments to perform ion exchange chromatography? So you can get them from Edvotek, www.edvotek.com. We will be able to, we sell this experiment on our website and also through distributors. All right, so let me just, actually, if one of my assistants, either Brian or Maria, if you want to post a link to the kit, that would be great. Thank you. All right, so I'm not going to get this perfect, but what you can do is you can see the samples here that I eluted. And you can see they're all, you can see the concentration gradient from dark to light, and you can see the red fractions versus the blue fractions. So in order to make this into a true STEM activity, we are going to incorporate mathematics. And luckily, that's easy with this experiment. If you have a spectrophotometer, they can take an absorbance at 550. So we actually did this in the lab at another time. I do not have a spectrophotometer at home here with me as much as I would like one. Basically, we know the concentration of the stock dye. So to make a standard curve, we are going to make dilutions of that dye and measure the absorbance. We create an XY scatter plot with the concentration on the x-axis and the absorbance on the y-axis. And then using Excel, we create a line of best fit. The equation of this line can be used to calculate the concentration of a sample from its absorbance. So if we plug in the numbers from sample 1 into this equation, the absorbance of 0.336 gives us a concentration of 0.023 mg per mil. Now, in manufacturing, we often want to figure out the percent yield, so how much of the sample we have recovered after we ran the chromatography experiment. There will be some that doesn't bind to the column and flows off early, and also some that sticks tightly to the resin and doesn't elute. So if we look at the column, let me take the column off. You can see the column still has a little bit of blue stuck in the frit and a little bit of red stuck in the column. Now, the process is not going to be 100% efficient. So we want to know how much we've recovered in our fractions, though. So since each fraction is about a mil, we can use the concentration determined to figure out the mass of dye recovered. So in this case, we've recovered the equivalent of 0.35 mg. So we actually started with 0.4 mg of dye. So if we calculate the percent, which is part of our whole times 100, we get 89%, which is not bad, considering the way this experiment is done. So if we were making a medicine in a laboratory, we'd be happy. That's a pretty good yield. So we would take these fractions, mix them together, and then go to the next step in manufacturing, which would be filtration or concentration or even another chromatography step. So we have two questions. The first is, what's a topic that's going to be presented on July 9th? Today is July 9th, and we're talking about chromatography. We have not yet decided what the topic for our next live stream is. But if you want to give us some suggestions, you can in the sheet, the Google sheet that we provided. Maria, if you could put that link back again, when you sign up to get the slides, let us know what you'd want to see next. You can use, another question is yes, we use spectrophotometry to determine the absorbance of these different fractions. So I didn't do that here. We did it, this is data from another experiment that was performed in the lab. But basically you'd make a standard curve using dye of known concentrations, and you would measure their absorbance. You plot them on the axis with concentration on the x-axis and absorbance on the y-axis. And then we are going to make a best fit line to give us an equation of roughly the relationship between concentration and absorbance. And so we would take the absorbance of each of the fractions. Once we have that absorbance, we can use the equation to go back to the estimated concentration. Now, things aren't going to be perfect. You can see our best fit line, while, it doesn't match every point exactly, and that could be for a lot of different reasons. Certain samples, things might have not been diluted properly. There are going to be errors in your measurement, but it gives us a pretty good estimate of how much dye was eluted or how much protein was eluted. All right, so at this point, if there are any other questions, I'm just going to run through, do a wrap up, but I'd love to answer any of your other questions, and you can get them in the chat box. We did have a question: can you reuse the resin? That's a great question. You usually can regenerate the resin. What you would do is run a really high salt buffer through it, which would elute the last of the bound dye molecules from the column. You might not get 100% perfect clean resin afterwards, but you'll be able to repeat the experiment, especially if you're using the same samples. Then you don't have to worry about cross-contamination. So we had someone ask for P-Glow when we can use protein electrophoresis to determine if contamination was present. What can be done with this activity to determine the quality of the lab technique employed? With P-Glow, basically it's going to be similar. So P-Glow, you would take the sample that you extracted from the bacteria, you would run it on a gel, you would have one band, your major band would be represented from your P-Glow protein, because you're overexpressing it, you're creating a lot of it in the bacteria, again using those bacteria like factories. And then you'll see smaller bands from other different proteins that are present. Now, with this activity, if we're talking about P-Glow, one thing that you can actually do is do an exclusion. You can do size exclusion chromatography of P-Glow. You can actually use a UV light to track the movement of the P-Glow through the column and see where it elutes. And then what you would do is take that bright glowing green column fraction and put it on a gel, and that would allow you to determine the purity of the purification. So how much you actually purified away from the crude lysate, so the cells that you just exploded. So if you ran the cells that you just exploded next to your purified proteins, what you would see is that you have an enrichment of your P-Glow protein, and then you have a decrease in the amount of other proteins in the sample. So you can see how we enrich for the P-Glow. Okay, so we have someone will be live again on 7:09 at 1 p.m. Eastern Daylight Time. So that notification, that was just in the information for this live stream. So I set up the live stream in advance. And so if anybody subscribes to our channel, you might see that. So it just would let you know what to expect. So when the presentation is archived on our website, I'll fill that in with relevant information about the presentation and the slides. So our wrap up. So I think this is a great experiment that really allows us to explore a lot of different aspects of STEM. The science behind the lesson, we learned about electrostatic interactions between dye molecules in the matrix. We talked about the chemical properties of proteins that give them charge and how this charge affects chromatography and how that changes when we add different solutions. So the way we can affect the properties of a protein just by changing the pH or changing the salt concentration. So technology plays a big role in biotechnology and in biological sciences experiments. For this experiment, we used bioinformatics tools to analyze a protein, in this case, Glorin, the protein I worked on in graduate school. And then we also used, if we were in the lab, we would have used a spectrophotometer to measure the absorbance of the samples. Chromatography is an engineering process in that we are designing and building a method to separate molecules by charge. And then we used mathematics to calculate the concentrations of our fractions based on the absorbance of the samples. And then we used that information to calculate our percent yield using a standard curve. So I'm going to go through some more resources that we have to teach chromatography. We think this is an amazing biotechnology technique that can be used in the classroom. And then also we can use it to teach in distance learning as well. So who knows what school is going to look like in the fall. We want to make sure to give you options. So for size exclusion chromatography, which is going to separate molecules by size, we actually have a free Edvotek at Home lesson on our website. This can be used for distance learning in that the students explore concepts through paper-based worksheets and exercises, and then apply the principles once you get back into the lab. Each Edvotek at Home lesson is going to include study questions and a PowerPoint presentation. And so someone brought up in the chat box, going back to our GFP question, it's really neat to run the native versus the denatured GFP side by side on a polyacrylamide gel. It's very cool because you can actually use the UV light to shine on the gel to see the green glowing protein. But then we can also see the effects of secondary structure of the protein, so how it's folded on the way it migrates through the SDS gel. So to go along with our chromatography at home experiments, we have video tutorials which detail packing the columns and performing the size exclusion chromatography. So paired with our Edvotek at Home lesson, with this lesson, those are great resources. And we also have some fun printable activity sheets, including a coloring page and a word puzzle to help your students visualize the process and learn the vocabulary of chromatography. And so you can actually have your students define each term in the word search to kind of reinforce the vocabulary necessary for electrophoresis. So we are also excited to be able to offer you professional development certificates for attending this workshop. So when you fill out the information form, if you haven't already, the link is in the chat box. So please fill out that form and indicate if you want us to send you a PDF certificate. And so this is going to signify that you attended the workshop live and have, taken part in a professional development activity. If you're watching this as a replay, sorry, we are only going to offer certificates to live attendees, but be sure to catch our next workshop here on YouTube. So finally, we are going to be presenting these slides and the presentation and the slides in the next few days on our website, www.edvotek.com. If you want us to email you when they're available, again, make sure to fill out that Google form and we'll contact you in a few days. Now, if there are any additional questions, I'd be happy to answer them. Chromatography is, a very fun process. It's easy to do in the classroom, especially when we're using these dyes. We're basically using dyes in salt water, so we're not worried about any chemicals that could be a problem with your students. It's easy to dispose of. We did have one person ask if we could regenerate the resin. So yes, you can definitely regenerate the resin. Again, using a high concentration of salt. You're gonna knock off all that dye that's remaining on the resin, and then you can use it again. So that works very well. If you're trying to come up with an activity to do from home for your students, you can pair these videos and these resources with a paper chromatography experiment so that they can kind of get the idea of how it's used in industry, but then actually do a separation and analyze it themselves. And depending on what your situation is, you could actually probably divide this kit up and set it home with students if they're only in the classroom once a week. If there aren't any, thank you, for the feedback on this presentation. I hope you enjoyed it. It's always nice to get the good feedback. I'm just really grateful that you were here to join me today to walk through this chromatography experiment. Believe it or not, it's already 2 o'clock. So we are at the end of our time together. If you need us, please reach out to us at info@edvotek.com or try one of our social media channels. We respond to YouTube comments, Facebook, Twitter, Instagram. You can get in touch with us any of those ways. Info@edvotek.com is our email. And then we also have 1-800-edvotek.com. And we really look forward to helping you get biotechnology into your classroom, into your distance learning situation. And so if there are any other ways that we can help you and you have more questions about how to create a curriculum around both in-lab and at-home experiments, you can email us at curriculum@edvotek.com, and we'll help you figure out the best process possible. So with that, if there are no more questions, again, we want to thank everyone for attending, and stay safe. want to see you back in the classroom this fall. So thank you very much for your time and have a great day.